the parameters to obtain parameters inside cells in terms of, for example, kinetic constants like K off. So in this case, we actually in a way simulate an experiment and try to adapt the parameters in such a way that it fits the experimental curves. Now, in case of membrane association, what we do is we photoactivate by TIRF material at the plasma membrane, and then look at the dissipation of fluorescence. And two processes take place. One is lateral diffusion in the membrane plane, and the other is the off rate. So we have two parameters that we want to fit. Now, because in a way that gets scrambled, Malte Schmidt, that worked on this project and that developed the cellular automata, had a really smart idea. We had the difficulty actually, because the lateral diffusion was so fast, to have enough dynamic range to determine the off rate, because it was way slower. So what he did, he basically applied a lot of pulses that you see on the left side, such that he saturated the material at the plasma membrane, and there's little lateral diffusion that you can see anymore, and so all the decay is now the off rate. Now, end of the story is very simple. If you express PD delta, there is no change in the off rate, which means you have a passive mechanism. If you change two residues, two positive residues to neutral residues, we can actually see an increase in the off rate, which clearly shows that this electrostatic interaction contributes to the off rate of this interaction. So what do you have a situation here? When KRAS is novo-synthesized, it gets prenylated, and it then basically distributes over all membranes. And so get dispersed. The problem is that it's very slow, so we have a kinetic barrier to actually get a concentration because of the electrostatic interaction at the plasma membrane. So PD delta traps a material that's cytoplasmic, concentrates it there, and then lets it move very fast, such as it can get retrapped at the plasma membrane. Is this now the end of the story? Because this is not really dynamics. No, it's not, because everything moves. The point is that even the plasma membrane is constant vesicle fusion and fission processes that mix up material. And that will actually lose the organization. So if you, for example, now do again a photoactivation experiment, very similar to the off-rate determination, and we now put, for example, an endocytosis inhibitor like Dinosaur in the system, we can actually see the shift of the off-rate shift towards the left. So basically it becomes smaller. That means that part of the process is really endocytosis that takes RAS away from the plasma membrane. So now this is basically a movie that tries to summarize these results. What we have is a constant battle against randomization in the system again. And PD delta constantly brings it back. So we have an active process of vesicle fission where negative charge as the endosomes matures is lost, and so you get loose binding. Now it will all mix on all membranes in the cell. PD delta recaptures it and explores the environment so fast that it can get retrapped. So again, it's a system that fights the tendency of the system to increase its entropy. Important part here is now if you take tumor cells, and these are pancreatic duct adenocarcinoma cells, and we look at the RAS distribution, again we downregulate PD delta. We see exactly the same effect of randomization of RAS on all membranes. So look on the left, you see very nicely now endogenous RAS. We detect it with triple RBD. Very nice plasma membrane distribution. We downregulate PD delta, and you see it on all membranes. So we really dilute it over all membranes. So we can interfere with that system as well. Now, I'm not going to show this because of time limitations, I presume. So can we really use this approach to actually affect oncogenic RAS signaling and affect the phenotype of cancer cells, and specifically pancreatic tumors? Because as I said before, 90% of all pancreatic tumors have KRAS mutations, oncogenic KRAS mutations. Now the first indication that this really works was shown by this experiment where we use a mouse model, so-called mouse pancreatic duct adenocarcinoma model. These are basically a cell model, where a single allele is replaced by allele that encodes for oncogenic RAS. This is a very physiological model for pancreatic tumors. It's actually sufficient to get complete transformation. Now, what we have done in these cells, we have downregulated PD delta and used clonogenic assay to look at cell growth. And what you can see very clearly over here is that we don't get a complete downregulation of PD delta, but there's a clear correlation between cell growth and the downregulation level of PD delta. Okay, so this is the normal situation. You see very lots of cells that grow. When we downregulate PD delta, we see very clear lower amount of growth. Interestingly enough, also cells that are transformed with HRAS do exactly the same thing. It's a universal factor to maintain RAS organization. Now, the more exciting result comes now. The problem here is that we're looking in a way at an integrative assay. We're looking at an assay after 7 days, we look how many cells are just there. What we really want to do is, are we killing cells specifically? Are they dependent on RAS for their survival? Or are we affecting proliferation? And the clonogenic assay in a way integrates these two parameters. So we need to look real time at growth on the condition of PD delta downregulation. So here we use electrical impedance as on a surface to actually measure the growth, the surface occupation of cells. So basically we monitor in time how cells grow. And we have here an inducible system, a doxycycline inducible system of shRNA against PD delta. So when you add doxycycline, you downregulate PD delta. Now look at the blue curve on the top. So these are KRAS-dependent cells. That means they are dependent on KRAS for their survival. Without KRAS, they are addicted to basically their oncogene. Very clearly, we kill these cells, and we kill them specifically. Okay? If we take a cell line that is independent of KRAS, we don't see this effect. There's no effect whatsoever. So we're really affecting RAS signaling, oncogenic RAS signaling in that sense. If you take cells that, for example, have wild-type RAS, it's exactly the same situation. No effect whatsoever. Now, this, of course, screams to find a small molecule to actually affect this interaction. And we have started a very nice collaboration with the Waldman department, which makes these inhibitors. And I'll show you just, I will not disclose the inhibitors, obviously, because the process is not that far, but I'll show you how we actually address this problem, or how we try to develop these inhibitors. So I go back to these imaging approaches to actually see if inhibitors can interrupt that interaction. So again, we look at the interaction between PD delta and in this case, Rib, is for a specific reason that I cannot mention because of time limitations. It's again another G protein that's oncogenic. When we add the inhibitor, now look at the alpha map. We see before we add the inhibitor, we see a lot of interaction between PD delta and this Rib, which is the normal situation. Okay? Then we add the inhibitor, and you see it goes to the blue, so we lose this interaction. Remark also, when you look at the top panels, the green panels, that we go from a soluble state to something that's completely membrane bound, but randomly. So we're really inhibiting this interaction. Now, the latest inhibitors, and Herbert, you have not seen this yet, but the latest inhibitors that were developed there that have a very high affinity show that this interaction inhibition is within 1 minute. You can see this very clearly over here. And this is really a very fresh result, where you can see that when we now apply this inhibitor to pancreatic, now human pancreatic tumor cells that are KRAS dependent or KRAS independent, you see the following. So in case of KRAS dependent situation, the PD delta inhibitor clearly inhibits the growth, and very strongly. Versus the control group just simply grows, as shown by the light curve. However, when we take KRAS independent cell lines that don't depend on KRAS for their survival, that are not addicted, then there's no effect whatsoever of this inhibitor. So the conclusion of this is that if you understand the dynamics of a system, the maintenance of spatial organization, you can actually rationally design approaches to affect the response properties of the cells and thereby affect their social behavior. I call this scrambling of the dynamics of spatially organizing reaction cycles, allows the downmodulation of oncogenic RAS signaling, and thereby they affect the phenotypic response of RAS-dependent transformed cells. So this is an institute-wide effort where different disciplines come together. So I've already mentioned Herbert Waldman as a chemical biologist that develops all these inhibitors, but also makes, for example, semi-synthetic proteins that allow us to actually nail the acylation cycle. We have also close collaboration with the legend Alfred Wittinghofer, who did a lot on the RAS structure, but all his, let's say, structural insight help us a lot to place it actually in a higher context. And the people that I've put here in red are the people from my group that were involved in this project, with a special attention to Hernan that actually developed these imaging approaches that allow us to look at this interaction and understand the spatial mechanisms. I thank you for your attention.